Thank you for joining us for this editorial panel discussion as part of Cell Press Cancer Research Week. I'm Deborah Sweet, your compere for today, and I'm joined by four of the editors from our wonderful editorial team. Jiaying Tan from Cell, Steve Mao from Cancer Cell, Nikla Emambokus from Med, and Sara Hamilton from Cell Reports Medicine. I'm going to let them introduce themselves, starting off with Jiaying.
Hello everyone, my name is Jiaying Tan. I joined the Cell team in 2013 and it's in my 7th year in the team. I'm currently a senior editor in the team. And Cell has always been at the frontier of publishing exciting biology, and the theme of the frontier of biological and biomedical sciences has always been at the heart of our publications and our interest. And it goes without saying that the cancer research and oncology research is a big part of it, and it would always be excited to publish the great work in that field.
Next.
So first of all, I'm very happy to be here to talk to our readers and viewers about our interest in clinical oncology. So Debbie, thanks for this opportunity. I started my editorial career in Cell in 2012, and then I joined Science editorial team in 2017. So in my editorial roles in both Cell and Science, I have always been very interested in the development and progress made in cancer research field, and also had the opportunities to work with talented scientists in the cancer field. So I'm very glad that I came back to Cell Press a couple of months ago as the new editor-in-chief of Cancer Cell. So in the last couple of months, I have been thoroughly enjoying working with old colleagues and new colleagues, and also exclusively with the scientists in cancer community. So Cancer Cell is a leading cancer and oncology journal and is often viewed as the first home for cancer researchers to consider submitting their papers. So we are very proud of that. And we understand that we cannot have this success without the help and support from our community. So we appreciate very much. And we'll keep trying our best to make the journal even greater.
Okay, Nikla?
Hello, everybody. It's a real pleasure to be speaking with you. I am Nikla Emambokus, and I am the editor-in-chief of the new Cell Press journal, the new Cell Press flagship medical journal, I should say, Med. I have been an editor for over 15 years and was previously the editor-in-chief of Cell Metabolism, and I have also been the executive editor for Cancer Cell. I have been passionate about scientific discoveries my whole life. Over the last decade or so, my interests have pivoted more explicitly towards human health, and I am super excited to be leading Med. With Med, I see the opportunity to do something different. Globally, we are at the golden age of science and discovery. However, the translation to human health and to clinical therapies is still slow. Getting the science to the clinic is an extremely complex process, but I don't think that it needs to be so, especially if we can get all the parties involved at the same table. So this is what I am hoping for Med. Bring together not just basic and clinical researchers, but also biotech, pharma, and regulatory scientists to look at today's medical problems together rather than sequentially. I think that cancer is a prime example of this. Immunotherapy has so much potential, but the actual clinical footprint is small. It's not just a matter of price costs, it's figuring out the science to make the system more robust and save more lives.
Thanks. It's a great goal. Sara.
Hi, everyone. My name is Sara Hamilton. Like my colleagues, I'm really happy to be here and to talk about my journal and some of the exciting things that are happening across cancer research and what we're doing at Cell Press. So I joined Cell Press about 5.5 years ago. I was a deputy editor at Cell Reports, and now I'm the editor-in-chief of Cell Reports Medicine. So like Med, Cell Reports Medicine is a new broad scope journal at Cell Press. So we're focused very much on advances in translational and clinical research. And the goal is really to provide a centralized forum where scientists and clinicians can find important developments in their field. Translational cancer research is my own background, so and it's something I'm very interested in, and translational research across the board. I think there's just really exciting things that are happening there. So I'm very happy to be leading this journal. Yeah, so.
Great. We're happy to
Oh,
have
and
you
I
here.
should say we're also open access. That is a very important part of us. So we are your open access translational journal at Cell Press, so gold open access. So that means anything that you publish with us, of course, is immediately and freely available for anyone and anywhere.
Great to remind everybody of that. Thank you.
Yeah.
Okay, so we'll start off with our discussion. So Jiaying, I'm going to start with you. You've been at Cell for quite a while now. So could you tell us a little bit about how you've seen the coverage of cancer research changing in Cell during that time?
Sure, Debbie. I would first say what's not changed. I think what's not changed is really Cell has always really in the position to publish the frontier of cancer research, wherever that frontier is. So in a way, I think the coverage of cell cancer-related content that Cell has put forward over the years really reflect where the field has evolved and how the field has evolved. I'll give you a fun fact. We were just in a strategy session the other day, and we pull out the highest cited paper Cell published in 1995. And guess what? That paper is about tumor suppressor P53 and its signaling relationship with FACS, right? I mean, that's a classic. And I'm trained as a cancer biologist, so P53, everybody knows it. But you can tell, like, that's what we were published and definitely proudly and happily publishing back in 1995 in the cancer theme. And in 2017, Cell published our first clinical trial. It's a phase 1 trial to use oncolytic virus to promote checkpoint blockade in melanoma context. So you can see that evolvement is pretty dramatic, even though put it in the lens of the progression of the field. I think it's quite understandable. So in a nutshell, I think it's really important for the editorial team in our way to sort of communicate with the community a lot and really get the pulse of where the field is, and really make sure that whatever Cell is publishing is representing that frontier, is both leading and representing where the field is going, and hopefully that with the kind of publication that we have and the visibility of our platform to send the kind of message of how to make sure the translational or the more basic end, as well as the clinical oncology community work together, because that's really, I think, the main theme or may change in the most recent years, that those two fields are coming together, and the word oncology research and cancer research used to be meaning very, very different things. Nowadays, not so much.
Yep, completely agree. It's changed a lot. It's also really fun to hear that P53 was, one of that top paper. It was cool. Okay, yeah, it is a classic. Yeah, Steve, you told us you're pretty new at the helm of Cancer Cell. So could you tell us a little bit about what kind of changes you're thinking about putting in place?
Yes. So again, Cancer Cell was founded in 2002, so more than 18 years ago. So we has always been very interested in publishing translational and clinical content, but they were still historically underrepresented. So we really want to make sure that we continue to publishing this inspiring, important, and high quality basic research content, but at the same time, increasing our coverage in clinical oncology. We are actually making a lot of changes to achieve this goal. So I just wrote an editorial detailing our major changes and asking feedback from the community. If you haven't seen it, I would encourage our viewers and readers to check it out. So here I will just detail 2 changes that we are doing that's specific to clinical oncology. So first, we are actively engaging with the community. So we have been talking with clinicians, and we will keep talking with them to talk more with this clinical scientists to understand their need, to better prepare us to serve them. We are also in the process of updating our advisory board members. So we are going to include a lot of clinicians and clinical scientists to represent the field. So we hope that by engaging with them, we can make sure that the community knows our continual commitment to publishing clinical content. So second is really why we think Cancer Cell is the best home to publish those clinical papers, because we are confident that Cancer Cell can provide a powerful and unique platform to bridge the gap between basic and clinical research. So we also often talk about, from bench to clinic translation, which has always been the pillar to advance modern science. Actually, just last year during ASH conference, a scientist told my colleague, Hamini Turk, her paper published in Cancer Cell several years ago has been the foundation of a drug that just got approved by FDA last year. So this is very good to know. But I would like to say that the other way is equally important. So basically the translation from clinics back to the research. So many clinical observations are extremely valuable to tell us about biology. So we will make sure at Cancer Cell, those clinical papers that we publish, whether it's a first-in-human proof of concept study or a clinical trial or a clinical investigation analyzing patient data. So we will make sure that those papers also provide value to our basic research community. So we will use our editorial skills and diverse formats to facilitate this two-way interactions.
Great, thanks. Really important. So Nikla, Steve's given us a great entry into sort of talking about the interface between clinical and basic research. And so maybe you can talk a little bit, you gave us a good introduction about MED already, but tell us a little bit about how you think MED will contribute to cancer research specifically.
Mm-hmm. Thank you, Debbie. Indeed, it is a first for Cell Press. With the launch of MED and Cell Reports Medicine, we are now squarely entering the field of medicine. Of course, this is not entirely new for our journals, as Jiaying said previously, and Steve before me. We have all our journals, most of our journals, I should say, have published clinical and translational papers before. However, with MED and Cell Reports Medicine, we are now, as I said before, we're squarely into medicine. With MED, this is what we live and breathe every day at the journal across all of clinical and translational research. So MED caters to a very broad audience across the whole spectrum of contemporary medicine, from head to toe, so to speak. Cancer is an important part of our scope. In fact, cancer has touched all of our lives in one way or another. As we speak today, my mother-in-law is being operated on in Milan for suspected pancreatic tumor. So for MED, my goal is not just to publish the most impactful papers in cancer research, but think about the disease in a more integrated manner. We can't just think of one disease in one silo, and MED's audience allows the best research in specific fields to be widely seen across different specialties. And we want the research to be widely seen and influence the thinking of others in other specialties. In a time of increased specialization, I believe it can be beneficial to be generalist. I think I heard the term specialized generalist before. I'm not sure where, but it makes a lot of sense to me. For cancer specifically, at MED, we are looking for cancer therapy papers that don't just move the needle forward for therapy, but also papers that inspire new avenues of research, even in different fields.
Sounds great. And obviously we wish your mother-in-law all the best for her surgery. That sounds, quite stressful, but great goal. And you talked about generalist, which is a great introduction to Sarah too, because Sarah's the EIC of a pretty broad scope journal. So could you tell us a little bit about how Cell Reports Medicine fits into the overall Cell Press family?
Of course, yeah, and definitely my colleagues have already provided a lot of the background here about the interest in translational and clinical work, or the increasing interest as fields move across Cell Press, and in particular in the field of oncology. And with MED and Cell Reports Medicine, as Nikla said, really advancing that and taking it to clinical work. So we are a broad scope journal. So we're interested in translational and clinical work across the spectrum. So cancer, of course, is part of that, but only part of that. And really, we're interested across. So if you, so really, as you take a basic discovery and think about how to translate it into something clinically useful. So an example of a Cell Reports Medicine paper would be something, you take your basic discovery, you verify, you translate it into different preclinical models to really understand the context or the conditions where this might work. Or you have a proof of concept study in humans, or full-fledged clinical trial where you've really taken what we've learned from the translational work and looked at what's happening in the clinic. And so we really are hoping to support what's happening across Cell Press and to really build this translational and clinical ecosystem. And so that is part of it. So also providing, you submit your paper in this domain to Cell Press and making sure that we have a venue for you to publish your work. And so that's also where Cell Reports Medicine comes in. And then it's also an open access option. As I said, this is a gold open access journal, and sometimes that is what you need for your work. And so having a strong open access option in translational and clinical medicine, I think is important. And so that's what the journal has to offer as well.
That's great. And you made a great point about us really wanting to make sure that all our authors who are interested in publishing with us feel like they have a home with us. I mean, that's really important to us. Yeah. Okay, I'm going to go back to you now, Jiaying. Tell us again, Cell's, you mentioned at the beginning, Cell's published some really great papers over the years, really great. But could you give us an idea of what kind of sort of more modern, the kinds of clinical papers that you're looking at now? Maybe tell us about one that'd be an example that you think is really good.
Sure. It's really hard to say like there's like one paper example, sort of the variety of types of clinically oriented work that that would be interested in. But I do have a couple of examples I'm happy to share with you guys. Like we published a paper from a group led by Ash Elizadeh in Stanford, I think last summer. It's a paper that basically deals with the question of how do we accurately predict a long-term outcome in the sort of oncology care space, like from a patient. And the paper was going into the element of, okay, it is not quite practical if you try to do invasive surgery or invasive approach to retrieve samples from patient at multiple time points and do like very comprehensive analysis. In the clinical setting, that's not very practical. So the paper comes in to say, okay, how about we think about non-invasive diagnostic? But the paper is very interesting, and I personally really love the paper, even though I didn't end up handling it because I wasn't in the team at the time, was because it takes the idea of sort of non-invasive approach, but also use the mathematical and statistical approach to design a dynamic process. And the concept coming out of that paper is that you pick different time frame from the patient over the course, get a lot of sort of information from the non-invasive measurement procedure, and use those dynamic information to optimize your predictive model throughout. So the reason I like the paper is that it clearly addressed a very important clinical question, which is a biomarker question, right? But it is not that I have a better biomarker than you, or I have a more accurate biomarker than you. It's really a different way to think of, okay, how do we move from a static point of view to a dynamic point of view and incorporate all those information that we can get throughout the time to optimize, to modify the performance and predictive power. So to me, that's a conceptually very different way to think of biomarker. And I do think that for clinicians and especially clinical researchers who study biomarkers and who have access to all of those samples, they could start think of, okay, how do I use that concept, apply that concept in my individual setting, be it prostate cancer, pancreatic cancer, or leukemia, right? So that's one type of the paper that I feel that I was really proud that Cell get to publish it, and full disclosure, our reviewers were also like really excited about the concept of the paper, and some of them are practicing clinicians. So I think those represent a type of the paper that we do think Cell is a good platform, because it really gives people a different thinking, both from the translational end and the clinical end. And another example I just want to briefly mention is we published a paper about tumor in situ microbiome in the pancreatic cancer. So it's basically to look at long-term survival pancreatic cancer patient and look at their microbiome in their pancreatic cancer, in their tumors. So it's not in the gut or in the skin or in other places, and try to look into how is that information informed of our understanding of those long-term survivors. It's incredibly rare samples, as you can think about. And there are caveats. I mean, that paper to me also represent another way that, it's not a Cell paper. It's like in this day and age, it's not just one type to say, okay, we have all the controls, all the best well-performed controls in the mouse setting, and we can draw a conclusion like iron tight in that context, right? When it gets to human, it gets complicated. But that paper really gave us some really interesting sort of lead in terms of, in the future, how do we think of the in situ tumor microbiome's role in the cancer long-term survival setting. There's a lot of questions. I would argue that the paper raised more questions than it answered, but that's exciting. And that gives people a clue from the patient basis to say, okay, those are the things that we should go maybe start look at. So, I mean, I'm just using those as two examples to showcase. They're very different, but I do think both of them appeal to a broad scope of cell readership.
That's great. The predictive ones, in particular, the idea that you can do that in a more dynamic way rather than just being static about it, so it would be continually adaptable, just sounds really neat. I mean, because it's obviously a big problem that we have to get better at, the predictive, using analytics in a predictive way, you know, using these markers. So yeah, really cool. Thank you. So Nikla, I'm going to go to you. So you've already told us that MED is breaking new ground for cell in quite a lot of ways. Could you tell us a little bit about how you plan to do things differently to help MED do that?
So, you know, I wanted to talk a little bit about bridging the gap.
Yeah.
There's the clinical and the basic research, the clinical, the bench to bedside gap. It reminds me a little bit of the London subway, where there's announcements saying mind the gap. There clearly is a gap between the exciting biological and technological sciences and the realities of the clinic. There's also a lot of hype going on. Curing cancer in mice is not the same as curing cancer in people. And how many times has a GP or PCP had to counsel the cancer patient that a purported new cure reported in a newspaper has not yet been tested in humans? So one concrete aspect that we are addressing at MED is communication. All MED research papers are clearly written and structured in a logical manner so that it is clear what has been done, how, what, where. The editors work with the authors to hone the message of the paper. Our papers include a limitations of the study paragraph, where the limitations of the work are candidly addressed. For example, is this a mouse study? I'm not trying to bash on mice, they are very model systems, but this is an example, or the limitations of the study could point out that the clinical sample size is still small, or that or point out that the sample is limited to particular ethnic groups, and ethnicity, for example, is a very important confounder, something that we don't often think about. And the limitation of the study paragraph is something that actually started at Cell Metabolism, and we received great feedback from our authors and our readers. People appreciated the open acknowledgement of caveats. Another communication aspect that we have as MED is that papers include a graphical, clinical, and translational scale. We call it the CAT scale. We built our CAT scale from the NIH framework for translational research. And the goal is to provide a graphic context for each primary research paper as to where it fits in the bench to bedside continuum. I think that this kind of information is important to enhance awareness of people who are not directly in the field, and also for lay people. So, and speaking of lay people, our papers include a context and significance paragraph where the work is clearly and very simply explained, but without exaggerating or overhyping. In fact, we call it the grandma paragraph, where we try to explain the research to your grandma. And I think that such communication stepstones help dispel a lot of misconceptions and make the complexities of bridging the gap from exciting basic research to actual therapy more real. And this is what we're doing at MED.
That's really important, because you're right, and we've talked a lot about how there is even within scientists and researchers, there's sometimes a communication gap. So if you can help cross that with MED, that'd be really great. So yeah, very important. So Steve, Nikla sort of alluded to this a little bit. We can talk about it a bit more. Reviewing clinical papers is a little different from reviewing basic research papers. And could you talk to us a little bit about what you think is important for reviewing clinical cancer papers?
Yeah, so definitely. So there are a lot of things that are different when you review a clinically oriented paper versus a basic paper. I think the one important component is this gap bridging. so Nikla just mentioned about. So for example, most of the cancer clinical or clinical oncology papers, their values lie within immediate potential to change clinical practice. But as I mentioned earlier, I think many papers also provide values beyond this improving patient care. So they tell you physiology, biology that cannot be get from mice, for example. So we think those papers are particularly valuable for cancer cell readers. So to review those papers, so we rely on our dedicated pool of knowledgeable reviewers. So many of them have both PhD and MD degrees. So they have extensive experience working in both basic and clinical settings. So we are confident that those reviewers will help bring out the biological insights from those clinical papers. So when we publish those papers, we can also accompany previews to highlight both clinical and basic values to maximize their impact. I think the other side of the coin is what Nikla just mentioned, mind the gap. I think Jiaying also mentioned it, because clinical papers are very different than basic research papers. So it is difficult, for example, to have perfect controls. So for those papers, for example, when you review a phase 2 trial, you probably require different sets of reviewers than when you review a paper reporting single cell profiling of tumor microenvironment in mouse, for example. So we are very mindful of that. So we are going to make sure that we work with the author and the field to develop unique and reasonable standards for different subfields. So the ultimate goal is to make sure that every author that his or her review process is a smooth, transparent, and productive. So we are here to help as much as we can to make the process as easy, short, and comfortable as possible.
Great. I hope you succeed.
Thank you. Yeah, I wish to.
I'm sure all of your authors will hope you succeed too. So Sarah, I want to move on to you, talk a little bit about negative results. There's a lot of discussion in particular how in clinical research in particular, there tends to be a pretty strong bias towards reporting positive results and not negative results, which is unfortunate because the negative results are important. So could you talk a little bit about how you're thinking about approaching that for Cell Reports Medicine?
Yeah, sure. So, I've definitely heard the same, and a look at the literature would support the focus on positive clinical trial results. And then if you also look at what's registered and never published, there's a lot done that's never reported, at least externally. And so I agree, like negative clinical trial results are critically important. But I think it's kind of a shift in thinking, both for the people who are actually conducting the trials and what to do with that. And then at the journal level as well, because it can also be hard to know, especially in basic research, I would say, how to interpret a negative result. But I think that's a little less complicated on the clinical side of this. So I think publishing negative trial results doesn't have to be a negative. I think they're effectively an opportunity to discuss what went wrong or to admit maybe that your hypothesis was wrong. And I think ultimately they do benefit your community and advance your fields. So Cell Reports Medicine is interested in those, certainly some of those, and see what's working for sure. Yes, positive clinical trial results are interesting and important. But again, also what's not working so that we can learn from that. So see how those negative results inform the next experiment in the lab. What Steve mentioned, you learn something about the physiology, and so that can go back and help what's happening at the bench. And then also how it informs the next clinical trial. So, is it an issue of recruitment? We have feline participation, patient recruitment, how you've analyzed your clinical trial, all the rest of it. So it can really help focus the next one. So I think journals should be a bit more open to these. And I think Cell Reports Medicine, our team, we discussed, and we definitely are interested in these studies.
Mm-hmm. Absolutely. And I think we all recognize the importance. I mean, Nikla, the first is not a cancer paper, but the first paper that Med published was also pretty much a negative result focused on coronavirus, right?
Correct.
Yeah. So, and it's important too. So it's online already. Okay, so now I'm going to get to like the fun part here, for the part that's the most fun for me anyway, and hopefully for all of you as the audience, is we asked all our editors to come prepared to talk to us a little bit about an area of cancer research that they think is particularly interesting or exciting right now, that they just want to sort of, you know, tell us about and tell us why they like it. So we'll start out with Jiaying, and I'm curious to see what you picked.
All right, Debbie. This is hard. This is really hard because like 7 years training in cell, I find everything interesting.
Yeah.
And the cancer field is fascinating, right? I'm trained as I did epigenetics, and back then there's a lot of thinking about how that as a different layer of genetics. So it's hard for me to say, comparatively speaking, one field is more interesting than the other. But since I mentioned a paper about, you know, tumor microbiome, let's just go with that theme. I do think it's one of those fields that is fascinating, and it's fascinating because it's new. There's a lot unknown. One could argue that, you know, it's almost sort of in comparison with the entire human microbiome field, not necessarily in the cancer field. It starts with a lot of descriptions, right? Lots of phenotypes, observations, and now we're getting into, okay, what exactly is going on at molecular level and the cellular level. In the tumor context, I think it's almost a mirror that, but a little bit step back, just because of the context and the practical difficulty and technical difficulty of ask the question. But I find it fascinating because it really bridges many fields together: immunology, microbiology, cancer biology, signaling, and sequencing. Right? When you think about like technical platform, there are certain reasons why certain questions in the cancer field was just not addressable before, because you can think of that's an important question. You don't have an approach to actually get to that question. So to some extent, I feel that that field really represents a lot of those different expertise people can come in to contribute. And as was briefly mentioned, I mean, clinicians also has a place, right? They are at the forefront, and they can collect those samples, and they know the clinical parameters and behavior of those patients, and upon treatment, and how are those connecting together, and how are this going to inform our general understanding of, like any human being. There's always this argument: how many human cells you have, how many microbiome cells you have, are you a human or a collection of microbiome? I mean, there's those kind of lay discussion happening in different platforms as well. So I think it is a field that embodies a lot of the crosstalk and the communication that really will help it drive forward. To some extent, it's the spirit almost for the entire clinical oncology and cancer research field these days. People really coming together with the samples that we can get from humans, and try to ask the question and use the experimental platform that we have to address those questions real time, like back and forth. So that's part of the reason that I find the field fascinating.
Yeah, another thing is the microbiome effects. I mean I'd be like, oh my God, another one, you know. Are there particular tumors that where it's a really sort of prominent part of the discussion, particular types of tumor, or just generally?
No, I mean, I think it really started with the gut, right? I mean, most people think of the gut and think of, like, even for different type, like melanoma and all those kind of, we remember there's a really beautiful paper published in Science altogether about looking at the gut microbiome and its relationship with checkpoint blockade response in melanoma context. But gradually there are more now, right? I mean, there are more, like what I was mentioning, tumor in situ. Like the paper that we mentioned was in the pancreatic tumor. We also published another paper that was more heavily on mouse model, but like look at the lung, like the microbiome in the lung cancer. So I think now there's going to be more that it just because of the level of granularity, we never get to see that. Like we don't even know what's existing. So I think it's one of the things that it will start with the description. It will start with the observation, but you have to start somewhere. But I do think that being able to at least see them and start to study them and how that communicate with the immune system, with the tumor, with the microenvironment, there's a lot can be learned about the dynamics. I mean, I've always find cancer fascinating. Maybe that's the wrong word. It's really devastating as well. But biologically speaking, scientifically speaking, it is fascinating. And part of the fascination is the complexity.
Absolutely, yeah. Something you've had quite a lot of contact with too, Sarah, the microbiome. Over the years. Okay, Steve, you're up next. Hello.
Yeah, so as Jiaying mentioned, this is a very difficult question because I cannot think of a better time right now to be a cancer scientist. Maybe not right now, you know, because of the COVID disruption. But overall, this is really, you know, exciting times for cancer biologists. So I would say that to me, one of those exciting areas is precision oncology. Because in the last couple of decades, we really have witnessed this technical revolution. So this allowed us to gather a lot of information from patients. So Jiaying mentioned one study. So those are non-invasive data collection. There are also invasive data collection. So we have all those data, right? Genome, metabolome, microbiome, and so on and so forth. So integrating this data to stratify biomarkers for precision treatment, especially in the context of therapy resistance, I think will have immediate impact in improving, you know, patient outcome in the clinic. At the same time, you know, goes back to the theme of bridging the gap. I think this also informs valuable biological insights that can be tested in the lab, and then to enable us to understand more of this complexity of the mechanism, and then to have better treatment. So I think many of these cancer researchers can be placed in this framework. So for example, it could be the new imaging technology and the computer algorithm, or it could be a large-scale single-cell sequencing of tumor samples. So all of those can be put in the framework of better obtaining and analyzing patient data. It could also be, you know, efforts to pinpoint signatures that differentiate cold versus hot tumors responding to immune therapy. So again, I really want to emphasize this theme of connecting clinical and the basic research community. I think those clinical studies will advance basic biology as well, especially teaching us the tumor resistance mechanisms. I think cancer cell is already part of this exciting development. As a matter of fact, our April special review issue is about overcoming resistance. So you will see from the 8 review articles, this precision oncology component is prominently featured almost everywhere. So if our viewers and readers haven't checked our special review issue out, and I would highly encourage you to read those very important review articles. And cancer cell, you know, we would really be happy to continue to serve and support our community to continue to be part of this exciting development.
Mm-hmm.
I would definitely second that. Those are beautiful reviews because I already read them.
That's great. You're giving me something fun to do at the weekend. That's great. Thank you. I should go read them too. Sarah, do you want to go next?
Yeah, sure. So this is where my own background comes into this. So I was going to talk about, I mean, it's what's not interesting. We're a broad journal and there's a lot that's interesting, but I was going to focus on immuno-oncology. It's something that we do hear a lot about, but I'm an immunologist and I trained in an immuno-oncology lab, so I still find this field fascinating. I mean, it's relatively new in the grand scheme of things, but the progress of the last decade has really been kind of incredible to watch. But where this takes us next, so it's conversations with Jiaying actually that turned me down this rabbit hole, but really trying to understand the long-term effects of some of these therapies, because they really are quite new. And you know, a lot of these targeted therapies are effectively turning cancer into a chronic disease that's managed. And so what we don't really understand a lot of the long-term effects of these extensive exposures to these agents, and in particular to some of these immunomodulatory agents and immune-related adverse effects, which we know happen. So really understanding, so evaluating them, monitoring how to manage them. So really understanding in patients the long-term effect of these and how we manage them. it's not, specific to immuno-oncology. Obviously, this is true across the board, but I think understanding the effect of these on patients' health over time is quite interesting.
Yeah, and also managing and controlling them appropriately to make sure that you do get the therapy right without, I mean, obviously cytokine storm has still been a bit of a problem. So yeah, yeah,
but yeah, relevant here too. Yeah.
Yes, absolutely. Okay, so Nikla, you want to go?
Right. I mean, there's so many areas of cancer that are interesting, right? But taking the perspective of med, where we are very much focused on therapy, I wanted to highlight the intersection of big data, AI, technology, and cancer. And there's so many facets of AI in cancer, from cancer research to diagnosis to care, all the way to the clinic. We have seen big jumps in the predictive abilities of AI in screening and in diagnostics. We're also seeing a lot of great data in drug discovery, for example, in epitope prediction and vaccine development, and the identification and validation of biomarkers under the area. However, the application of AI to cancer seems to be more in fits and starts than in a concerted and integrated way. So if we go back, starting in cancer research, I am looking for more systematic methods of mining all of the omics data that we already have, and a way of dialing down the noise level that's coming from data heterogeneity so that we can yield meaningful leads. For diagnosis, I'm looking at a seamless integration of AI with portable devices and wearables. For example, using a phone app to screen a suspicious mole rather than waiting for your next dermatology appointment. I mean, the aim is not to replace the physician or contribute to overdiagnosis, but proactively recognize early warning signs. Another aspect that seems underutilized is predicting synergistic or even antagonistic combination therapy responses at different time points, and integrating them with electronic health records and wearables to get a more holistic view of the patient. True personalized precision medicine has happened in pockets, but it's not yet part of the standard of care. And the application of AI in cancer can definitely accelerate this.
We've seen a lot of, been interested to see the things that have gone on already in terms of using AI in radiology and really supporting radiologists and helping back up those diagnoses.
Yes.
And even be a first level screening. Yeah.
Definitely help decrease the pressure on the radiologists.
Yeah, and help them consult with a broader range of images too, because there are many more than they could possibly see themselves. So yeah.
Exactly.
Yeah.
And at Med, we are eagerly awaiting the next steps, the next stages of integrating all this data and being able to make better predictive predictions, for lack of a word, for what. Trying to decrease the rate of mortality from cancer.
And that's a theme that's come up in several of these conversations. I mean, Jiaying talked about, using biomarkers in a predictive way, and Steve talked about prediction too, and, precision oncology and stuff. So it's, it's a common theme. So I can see why you're focusing on it. I know that was a hard question. I mean, you're right. We all become editors because we love science and we love all types of science, and it's really, really hard to pick something. But it's also really interesting for me to hear from you about the kind of things you're finding interesting at the moment. So thank you for taking the time to do that. And that brings us to the end of our panel. So thank you everyone for joining us as well. I hope you found it interesting. I hope you've got a better idea than you had at the start about these different journals in our ecosystem and how they fit together. And I hope, our editors feel free to get in touch with any of our editors if you'd like to talk to them more about their journals or even think about submitting. Thanks. Bye-bye.